pregunta o algo, sobre todo te la haga el micro, porque si no, no quedará grabado en el Zoom, pero la gente de casa no lo oye.
Perfect. Muchísimas gracias.
Venga.
Okay, everyone. Shall we get started? People from the internet, I guess they're listening, they're seeing the slides, which is the important part. Okay, I'll make a very brief introduction because we need to devote time to Pjotr and Arun, who very gladly are here today. I think there's a lot of things we can learn from them, and this is a golden chance to interact with people that really put out there open source tool and develop solutions for real world. I think this is our golden chance to take everything that we do and translate it into real applications. Okay, so there's a brief introduction. Pjotr is a bioinformatician, as he says, at large and associate coding professor at the University of Tennessee. But for me, what really matters, and this is my biased opinion, is that he is the man behind many widely used tools, developer and maintainer. For what that means, that is all day attending tickets, attending complaints, as he likes to say. Very famous tools like BCF library, Samba, and the recently published PGGB. I think this is very important, and that's a really very important contribution to the community. For you to understand how he committed he is to open source development, he is founder and editor of the, I'm going to read it wrong, I know, Biohacker Archive, and a former editor of the Journal of Open Software. Open source software. Okay, he's going to talk about pangenomes, which is a very hot topic, and I believe we also have a very keen interest on understanding pangenome, the challenges, and all that we can do to accelerate this revolution. We also are very happy to have here Arun. He is a postdoc at the Department of Genetics at the University College of London, and he is what we usually call a ninja because he's a true life hacker. He codes, I mean, very well, and he's the kind of guy that takes out you from a problem or situation. Okay, thank you very much for being here today. He's going to talk us about common workflow languages and ccwl. Okay, so thank you very much for being here today, and the floor is yours, guys.
Am I clear? I suppose so. Yeah, so Eric Garrison was here a year ago, right? I think some of you have seen his talk. I'm going to do the dumbed down version, essentially. Yeah, so we'll repeat and we're in, and we'll discuss afterwards and try to clarify as much as possible what we're doing. So I'm an associate professor at the University of Tennessee. I'm actually based in the Netherlands, so I'm in Europe. Yeah, so I take remote work to an extreme. And I'm partially to writing tools, as Santiago just said. Yeah, so, and these tools are running, everywhere. They're all in all sequencing centers, essentially. Yeah, so, I mean, Bioconda is just getting close to 1 million installs for the different tools. Yeah, so, but what I want to talk about is the future. These are the ongoing things. So we have an NSF grant to build a RISC-V supercomputer. This is based on the Black Parrot, which is 500 cores on one die, essentially. It's a very nice project, with a different architecture altogether. We have another grant we just won with Eric for $1.5 million, and this is just to write software. Yeah, so this is a software grant. Try and get a grant like that in Europe. This is why I'm in America. And of course, we have a collaboration ongoing with Santiago, in the Wavefront, and this is the paper with Santiago and Eric. Yeah, optimal gap affine alignment in OS space. So the take-home message, in 9474, somebody wrote that Agatha Christie at 84 is still getting away with murder, right? And now it's 2024, and the reference genome is 20 years old, and we're still getting away with murder. Yeah, and I'll try to explain that a little bit. Yeah, so this is the mapping slide. So this thing here is called the reference genome, and it's notably linear, right? It's just a line. And when you get reads that come from the sequencer, basically what people do is they align it against this reference. Okay. Yeah, you probably all know about DNA, right? See the 4 letters and all that. So this is a simple explanation. It's complicated, and as Santiago can also explain, when you do the alignment, you have to deal with errors in the reads, and you also have to do with highly repetitive regions, that look very much the same on the reference genome. So sometimes the read will fit in different places. And if you use the standard tool BWA, I think the current strategy is to position it randomly. Yeah, and non-deterministically. That means that if you run BWA 2 times, you get a different result, which is kind of annoying. So how many people are in the reference genome, right? Essentially 11 individuals. Yeah, so there's 11 people that made up the reference genome, and they're all from a very small part of the US. And there's one person in there who had contributed 70% of the reference. Yeah, and we think he was a mixed African American. And then 7% was added later, from 50 different sources. Yeah, because there were parts missing in the reference genome, and they tried to make it better. So 20 years later, all the tools that I use today, I would say 99% of all sequencing centers are based on the reference genome. Yeah, and if you take all the DNA that is sequenced in Africa, and there's a paper about this a few years ago, 10% is not on the reference genome. Yeah, isn't that stunning? Yeah, and it does not mean that if you take a random African that they will miss 10% of their DNA, more like 2% or something like that. But if you take all the DNA that was produced in Africa that we have, right, you miss 10%. So it doesn't map against reference genome; it's just missing. And the other point really is that if you look at a populational variation, between individuals, a reference genome cannot really capture that, right? As if it's just a linear genome. And this has real implications, because if you go to your doctor, and the doctor tells you, well, I'm afraid you're going to get, a heart attack in 5 years' time, right? I mean, this is what we're heading for. If it happens that you're, not well represented in this reference genome, can you trust that analysis? And the answer is no. Yeah. And what people have been doing over the last 10 years is that they created their own reference genome. So you got a Japanese reference genome, so Brazilian reference genome, a Spanish one perhaps. But if you have all these different reference genomes, and you start to compare results, it gets very hard. It gets very complicated very, very quickly, right? So this has implications, directly also for cancer, immunotherapies, and all that. And that's why, it's a murder, and we're getting away with it. So why did we get away with it, in a way, right? So why did it work? And the point really is that we are pretty much alike. Despite we think we are very different from a population genomic point of view, we are very much alike, which is cool. And this is because humanity was always wiped out 70,000 years ago. We can see basically from the variation in the genome, right? We can trace it back, calculate it back. So we went through a very small bottleneck, probably a few thousand individuals. Yeah, and since then, everyone has grown out of that. So the reference genome kind of works because we're actually very similar, right? So we got away with murder for some time. But other species have far more variation. Even if you look at the mouse, the mouse people for a long time have known that their reference genome did not work. Yeah, I could use a stronger term, but it was quite useless. Yeah, so and the mice are the victims here, right? And also all the other species that we've been working on. Even if you look at humble bacteria, yeah, or viruses. And what is interesting that people working on these smaller genomes for bacteria and viruses, they actually had already a pangenomic approach before we even thought of it. Yeah, because they had no use for a reference genome. So just to try to explain what the pangenome is. The pangenome is a new approach which replaces the reference genome driven approach. Yeah, so you take a population of sequences. So every colored line here is one individual, right? One human. And if you can create a graph of the variation. So basically, if I would sequence Santiago and I would sequence myself, right? where we are the same, you're a node, right? So the sequences are basically overlapping. But when you differ, you go separate, right? And this is how you can build a graph. And if you put a third individual, you do the same thing. So the nodes, they are shared, right? They're the shared sequences between individuals. So we build a graph, this is one representation. And you can see that, there'll be, for this brown individual, right? So the brown individual goes this way. We call it the path, right? And it's separate because there's a SNP variant. There's a G instead of a C. So all the others have a C here, right? This one has a G. Yeah, and we call this a bubble. This is just a terminology that we use. And this is a simple explanation, but what is cool about it is, first of all, you can represent very complex variation this way, right? So you can get bubbles within bubbles, like we have here, inversions, deletions. Yes, so you get a much better view of the actual variation in that population. And then we can use this graph, to align the new sequences, the new DNA that comes from the sequencer. So every read we align against the graph rather than aligning against a reference genome. And there's quite a few studies now that prove it's a superior approach. There are challenges though. We're aligning every sequence with every sequence. Yeah, so this we call, Eric called this an unbiased approach. Yeah, so he doesn't want to rely on the reference genome in any way. Yeah, so we have competitors. There's two tool chains that have been produced by now. One of them starts with the reference genome and then builds out from the reference genome the graph. But if you do that, you still have some bias in there, right? Because the graph is sort of pointing towards the reference genome. So he takes an unbiased approach, and this has led to the PGB Nature paper that we got 2 weeks ago. But the key thing is that you have to align every sequence with every sequence. Yeah, so 70 humans, which was the human pangenome reference that was produced last year, can be done in 2 days on our 11-node, 24 CPU, 2064 real cores computers, right? We're using network distributed storage. Yeah, so we can do it on the cluster, and basically, but what we do, we do it by chromosome, kind of. Yeah, and Arun will talk about that. He's going to build an Arabidopsis pangenome for you. And one of the problems we have is that the current tooling assumes that the whole graph is in RAM. Okay. So why all to all? The title is not very visible this way, but yeah, all to all is basically because we want unbiased graphs. So if a sequence comes in, right, we want to place it somewhere on the graph, and we really cannot anticipate where it goes. Yeah, so you end up doing an all to all. It's a quadratic challenge, and it's going to get really interesting because sequences are going to be very, very common, right? We're getting to the $100 whole genome probably next year already. Yeah, so that means it will be in every hospital, every clinic, everyone in every interest in any species will be sequencing at this, at these prices. So I'll continue, but I think it's time for Arun to do his thing. Arun, do you want this mic? Or you can use this mic also, I think. Maybe it's easier because you're sitting. Try and say something.
Hello.
Is it clear online?
Yeah. Hello? Yeah, I think it's okay. Maybe it should be a bit closer. Yeah. Yeah, I like that one actually. Hello, can you hear me? It's not on. Yeah, no, let me switch it on. This one. Small. Okay, yeah. Hello? Yeah. So yeah, So I'm actually going to talk about the software that we use on HPC, on the workflow languages, package managers, and workflow language implementations that we use. So our HPC stack basically. First, quickly, who am I? I'm a postdoc at University College London. I work in bioinformatics, but I'm really a computer scientist. I just switched to bioinformatics very recently. I'm very fond of the Lisp programming languages and domain-specific languages and novel computing paradigms. I mean, that's really the thing that I do. Yeah, I just happen to be working in biology because it's a nice place to apply these things. So the stack that I will be describing today are these 4 tools. So CWL, which is the common workflow language. And ccwl is a domain-specific language that compiles to CWL. And ravanan is something that actually runs the CWL workflow. And geeks, which is our package manager. So I'll actually give a live demo of this. So first, what is the common workflow language? It's so maybe you've heard of Nextflow, SnakeMake, and there are tons of other workflow languages. CWL is a bit different from them. It's actually based on a standard, on a spec, rather than on one specific implementation like Nextflow or SnakeMake. And the workflow description itself is a YAML file, so it's easy to parse and easy to generate. As long as you follow the standard, the spec, you can implement your own CWL runner and it should be compatible. So hence, it's the common workflow language. It's also a fully statically analyzable in the sense that even before you run it, you can you know what steps are in the workflow and which has to come before the before other steps and so on. So you know the entire structure of the computation, if you will. This is not the case with other workflow languages, which are more dynamic. So this is really important because you can visualize the workflow as a graph, and it's easier to do type checking and be sure that your workflow is bug-free before you actually run it. Imagine writing a bash script on all the housekeeping, for file housekeeping and temporary directories and input-output files that you have to manage yourself. That's a real pain. CWL takes all that out for you and automates that in a very reliable way. So ccwl is our is a DSL that compiles to CWL. I'll actually show this in the demo. Yeah, let's get to the demo. I'll show you some real code. What's the question? No? No. This one. Okay. Yeah. Yeah. I joined the meeting late, so maybe. Yeah, this one. So this is it's a toy workflow that I like to demonstrate. It's a spell check workflow which just uses basic tools from core utils, things like tr and sort and all the Unix commands that you might be familiar with. Let me first show a picture of the workflow. So I can actually compile this to the .graphis language. And then I put it through dot. So this is what the workflow looks like. So you can see that there are 2 inputs. There's a text file and there's a dictionary. A dictionary here is just a list of correctly spelled words. Each one of the yellow boxes in between are steps of the workflow. Each one corresponds to one command essentially. And you go through some sequence of commands, and then you end up with an output. So the code for this looks like, here you go. So maybe I should split the screen. Yeah. So you can see that there is a a nice one-to-one correspondence between the workflow and the code. So the split word step you have here, and then here is the command, the tr command that it literally replaces spaces with a new line, I think. Yeah. And then you downcase it and sort the words and find the difference between the words in the dictionary and so on. And oh, this is not good. Do this. And here for in this final paragraph, you actually specify the connections between the steps. So so there is a long sequential arm to this workflow. So that is specified here in this expression, and then the other arm is in this expression, which has the sort dictionary, and then you connect them with other operators. So it's a really short and it's a really short description that can fit on a fit on a page, fit on your in your head, and maybe even on a T-shirt. So let me now actually compile it to CWL code. So what you saw is the concise CWL, the DSL that we have. Now I compile it, and you get the YAML, which is the CWL. You can write this by hand. I mean, the spec was originally meant for humans to write, but I mean, it's kind of long, so that's why we made this DSL that can just treat CWL as a compilation target. And now we run it. So we yeah. So here's how we run it. We have the CWL file here and an inputs file. So I'll show you the inputs file. It's just a JSON file which specifies. It's a JSON dictionary which specifies the inputs. So so everything here is fully typed. So you have the text file and and this thing called a class file, which indicates that it's a file type. There are hashes, paths, and so on. So let me actually run it. Yeah. So you can scroll back up, and you can see that the individual steps here. So yeah, this is one step, I think. Completed. Yes, up to here. So this is the short dictionary step, and it you can also find the path to the logs of the command in case something went wrong. That's surprisingly hard to do in many batch systems. It's a real nuisance when you're running things on the HPC for days and you can't find something goes wrong and you can't really find the logs and you have to keep guessing at the problem. But here you can just look at the logs. It's very easy. So yeah, so you see exactly. Oh no, it's small. Put it down. So you can see exactly what command was executed. Yeah, maybe that's a little better. So it's a sort command with the unique flag, and you can see what file has been passed to it and what file was written in the output. And the interesting thing here is that we actually use geeks to provide very strong caching. So the output is stored in a directory we call the store, and you can see that. Let's put it 3. you can see that there are these long hashes in every the file name. So if you change any one of the inputs, or even if you change the version of the tools that you're running, this hash is going to change. Or even if you change the compiler that was used to build build some tool like core utils, for example. If you change GCC, the version of GCC that built core utils, you'll have a different hash. So this is all this is provided by Geeks, the package manager, and we just use those hashes as an addressing mechanism, really, to name these files, so that when you run a HPC workflow, you're absolutely sure that that you have exactly what you think you have. Like usually on HPC, people run workflows for days, and then they put all the parameters and tool versions into the file name, and then maybe months later they write a paper and hope that they actually did what they are describing in the paper. So that's a real nightmare, and I didn't want to have that nightmare. So I wrote this tool that that provides strong caching, and you never run the same thing again. So on the last day when you're writing the paper and just before submitting, if you have this store, you just rerun the whole thing, and it'll pull out everything from the cache, and you can be sure that it's exactly what you think you have. So that's that. Oh, I'm also supposed to be showing you an actual pangenome. So here's one. Yes. So this is an actual pangenome that I'm running on an actual HPC machine. I've logged in on by SSH, and I'm going to run this now. So it's a pangenome for the chromosome 1 of Arabidopsis. Arabidopsis is a popular model plant. So this will take a while to run, actually. We can go back to the presentation. Yes, I should tell you about this. Yeah. Oh, yeah. So the main benefits of using Ravanan is that you get caching, which I told you about, and you have a clear one-to-one map from the CWL steps to Slurm to actual Slurm jobs that run on the HPC. Yeah. And we also have a higher cluster utilization than some other implementations, because CWL has these array objects, and the way we have implemented them, you can actually start computing on some elements before the entire array is fully populated. So we use propagator networks to achieve that. That's a separate topic. I don't really want to go into that in this talk. It's a talk on its own. But I do encourage you to read about it in this book, Software Designed for Flexibility. Maybe you've heard of SICP, Structure and Interpretation of Computer Programs, by Sussman. So this is from the same authors, and there are some really interesting novel ideas about for programming languages and different kinds of computing that we could do. Again, we use something called G expressions, which provide all the hashes in the that I showed you. So G expressions were developed by Ludovic Cortes. He's the founder of Geeks. This paper is also worth checking out. Yeah, that's it. Let's see if the pangenome is building. Yeah, I can take questions now when we wait for the pangenome to build.
I do have a question, because talking, I mean, this is a very uneducated question, but okay, I'm going to make it. Coming from a guy that is usually scripting everything in Bash or Python, bad, I know, and is putting together all his workflows and pipeline, and then just throwing it to Slurm, defining dependencies on a Slurm, and just offloading everything to a Slurm. I was very puzzled when you're assessing that using this language, your tool, I would get more utilization that, for example, the regular Sloan scheduler between my jobs. How is that possible? Because that sounds really interesting.
Yeah, because so you get better utilization because you have a better description of the workflow, really. So since you can fully statically analyze the workflow, and you have kind of decomposed it into smaller components, you can know ahead of time that certain steps only depend on other steps, and you don't actually have to wait for something else to finish. So so usually when people do it manually, they would build a pangenome from all the assemblies from the sequences, completely wait for that to finish before even starting the next thing. It's a manual two-step process. Whereas here you can add your analysis to the workflow pipeline itself. So the language interpreter knows exactly what is required, and it can actually start things as early as possible. So it's really a matter of organization rather than a matter of high performance code or anything. It's just better organized. That's it.
That's an awesome contribution because I mean, we all understand that reproducibility is one of the I mean most overlooked things in this field in particular, but in general in many fields. So I take it for granted that when you ship one whole pipeline, you ship it in this language,
Yeah.
and the user can download this and it's ready to go.
Yeah,
He can
yeah.
just push one button, as we usually say, and,
Yes.
and get it done. That's a very nice thing. That's a very nice contribution. Any questions from the audience or people over the internet?
Yeah. So, and what is also interesting about Arun's approach is that because you can basically start early or complete computations, in certain channels of your whole workflow, and this is really important in the diagnostic setting, right? Because we're getting sequences from the sequencer, right? And it gets pushed into the workflow, and people eagerly await whatever comes out first, right? You don't have to wait for the full thing to start work. All right, so can I get my slides back? Is it, or do I do it here, or you should stop? That's where, yeah, that's it. Right, gotcha. And now, Zoom meeting. Yeah, I should probably not push leave, right? Yeah, thank you. Yeah, so Arun's stuff is, there's a Git link there for Arun's stuff. His software is all developed online, of course. So I'm just going to continue a little bit with where we are going with this. Yeah. We lost the Zoom. Oh. Ah. That one. Yeah, it's good enough, I think. Yeah, this will work. I've chosen a large enough font. Yeah, so until pretty much today, most of the alignments that we do are based on k-mers, which is basically fixed size chunks of DNA. So you get a sequence from a read from the sequencer, you chunk it up into pieces, fixed size, maybe 9 base pairs or 15 or whatever, depending what you do. And the tool that we've been using very successfully, it was published in 1990, right? It's very commonly used by biologists to look in, if you have a sequence, to see in what other species it exists, or what other areas of the genome it exists within a species. It's called BLAST, but BLAST has a k-mer approach, and now Eric is doing the all-to-alls. We're already finding that we are seeing things we cannot see with BLAST. Yeah, so that's amazing, and they actually had some findings recently. So, but of course, doing an all-to-all is quadratic. So 500 genomes. So they're trying to do all the vertebrate genomes. So everything that has a backbone, like the mouse, the cow. Doing 500 of these different species. We've been trying them on Sarah, which is the Dutch supercomputer. They gave us 100,000 hours. Eric just used it. We also have been running on Frontier. It was just usurped, right? I mean, he's like a black hole for CPU hours at the moment. But we didn't complete, right? But it's all kind of experimental at this stage. So Eric's current requirement for 500 genomes, he says, is he needs 2 to 5 million CPU hours, right? So if Barcelona can help, that'd be very, very interesting. We probably need 10 million hours to complete, right? Because we're still tweaking stuff, and but with the $100 genome, we bring it to the clinic, and if people start, want to build real full pangenomes of their population, and you have to, account for 1 terabyte of data coming out of every sequencer every day, it gets kind of, interesting. So we also, we need to think hard. Okay, are we doing the right thing? Maybe we should improve it. Should we use some type of machine learning to predict these alignments, to prevent doing the expensive thing? Yeah, we've also been working using LLMs, using DNA as a language, which is, there's a few groups in the world now doing that, trying that. It's not very, I mean, it's experimental, you could say, right? Because large language models obviously have been optimized for natural language processing, and there's structure in language, right? There's grammar in there, which it's using, and we don't have this. We have, if you look at the pangenome, we have ideas that are in the in DNA that we can capture. So maybe the large language models have some, can capture that type of relationship. But it's work in progress. We've got one pre-published paper on using the large language models and the pangenome to work on differential privacy. Yeah, I think there's people working in the BSc also on that, and we met someone. But yeah, it goes a bit far to discuss all that now. Maybe we can do it after. Yeah, so we are in the intermediate phase at this point, where we actually have people running their traditional workflows using the reference genome, right? And we have alternative workflows which are using pangenomics. So we made this very nice and noisy slide. Yeah, so you have the sequencer here, right? So the upper line is the traditional way. So this is the traditional workflow that's and then it goes to this type of toolings, and we show that the results with pangenomes are different from the results that we have with the reference genome. And when you get those results, you can go back to other tools that that all still reference genome guided, and you can study whatever you're doing. And then for pangenomes, we have called this whole alternate world, right, which is the blue line, and it goes down here. So you get these type of analysis, which are leading to new insights. This is one paper that we had, and then we have again different tools to look at the output of the pangenomes. So this, I mean, people are using the top layer, the top level, but they're not using the bottom layer at this point, most of them, right? And what we want to do is bring these worlds together. And one of the crucial things here is the VCF format, which most of you will encounter when you start to deal with genomic data. It's a variant calling format. It's not perfect. Let's see. But they're creating a new extension for pangenome type, because the VCF format itself is also dependent on the reference genome, right? So if you project the pangenome on a VCF format, by definition, you lose a lot of information. So they've created a new extension for this format, and we are kind of keen to bring these we basically have to update these tools to understand the new VCF format and these tools to understand the new VCF format, and then see how they can, be useful to people. Because what you really want is this turnkey solution, right? Because most biologists, they don't care, this is for them, it's gobbledygook, they don't care. Yeah, but they want to see the output. Yeah, the other thing we're doing, and we will discuss this, currently everything in pangenomes is done in memory. So, and that was, just because it was the easiest thing to do at the time. But now we're thinking more about doing, there's two terms for it: implicit graphs or lazy graphs. I prefer lazy graphs. If you do functional programming, you have, lazy resolutions, right? So only code gets executed when it's needed. Yeah. And the idea here is to essentially do an all-to-all alignment, but you just build an index of all these sequences, so that you approximately know where they are, sitting in the overall pangenome. But we don't build the pangenome itself. Only when people start to zoom in on a certain section, you build a sort of a mini pangenome on the spot. Yeah, so this is the concept that we're now aiming for. And if you think about, I mean, we have problems with 500 individuals, but if you get to 1,000 or maybe 1 million in the future, you really want to have some type of data structure that you can just pull data from and say, okay, I'm interested in this area, right? And it will also scale better on HPC, of course, because you can distribute it easily. So what it means is that we have to rewrite the whole software stack again. And that's exactly what is happening today, right? So, but now it's based on the implicit graphs or lazy graphs. Yeah, but we still also have to discuss, and see if we can introduce new data formats. We're doing it with the Cornell folks, right? We're working on one. And also, if we can leverage different architectures, the GPUs, the TPUs, and all that. Yeah, and they're building all these nice data centers for large language models, right? I mean, essentially, investors are pouring billions of dollars into building these type of things, and they're going to be pretty useless pretty soon. So, I suspect we're going to use them. Yeah, so this is the wide range of collaborators that we have. Obviously Eric is the resident genius. Arun is here. We work on rat and mouse pangenomes and human, obviously. We have the NSF project, which is the supercomputer. We have NIH projects. We did the exercise with Sir Sarah at NWO, and now we're here in Barcelona for the Elixir Biohackathon. Yeah, so that's why we're here. Thank you.